To better understand the concept, let's start by breaking down the structure of biological molecules step by step, beginning with carbohydrates. Look at this model of a carbohydrate molecule. It represents a polysaccharide, a large carbohydrate made by joining many smaller sugar units, called monosaccharides, like glucose. The diagram shows a branched polysaccharide, where extra chains branch out from the main sugar chain. This type of complex, branched structure is commonly found in storage carbohydrates such as glycogen and amylopectin, which is a component of starch. Carbohydrates consist of simple soluble sugars and complex substances such as starch and cellulose, all composed of carbon, hydrogen, and oxygen. Glucose, a common simple sugar with the chemical formula CHO, typically exists in a ring structure and can link to form disaccharides like maltose. Monosaccharides, including glucose and fructose, have a single carbon ring, while disaccharides like maltose and sucrose contain two rings. Both types dissolve readily in water. Larger carbohydrate molecules such as glycogen in animal cells and starch in plants serve as storage forms of glucose, with glycogen and starch composed of long glucose chains. Cellulose, with even longer glucose chains arranged into microscopic fibers layered to build plant cell walls, along with glycogen and starch, exhibits low water solubility. Electron microscopy reveals that cellulose fibers in plant walls consist of numerous interconnected long-chain cellulose molecules. Monosaccharides are the simplest form of carbohydrates, functioning as the fundamental building blocks, or monomers, from which larger, more complex carbohydrates are constructed. As illustrated in the image, common examples include glucose, fructose, and galactose. These molecules are often referred to as simple sugars because the term monosaccharide literally means single sugar. They represent the most basic units of carbohydrates and cannot be broken down into simpler sugar components through hydrolysis. Their simple structure and essential role in energy metabolism make them vital to numerous biological processes. Here, the diagram shows how maltose, a disaccharide, is formed by joining two glucose molecules, which are monosaccharides, through a process called a condensation reaction. First, two separate glucose units are shown on the left side. In this reaction, one glucose loses a whole hydroxyl group and the other loses a hydrogen atom from its hydroxyl group. Then these two parts combine to form a molecule of water, which is released during the process, as shown in the diagram. After the water is removed, the remaining parts of the glucose molecules join together through a new bond called the glycosidic bond as marked in red in the diagram. This bond links the two glucose units, forming maltose. In short, the diagram explains how two simple sugars combine by losing water to create a more complex sugar, maltose, connected by a glycosidic bond. So, we can conclude that a disaccharide is a type of carbohydrate made when two monosaccharides, simple sugars, join together through a condensation reaction, also known as dehydration synthesis. During this process, a molecule of water is removed, and the two sugar units are connected by a covalent bond called a glycosidic bond. The image gives three clear examples of common disaccharides. Maltose consists of two glucose molecules, both shown as identical sugar rings in the diagram. Sucrose, also known as table sugar, is made from one glucose and one fructose molecule. The glucose appears as a blue hexagon, and the fructose as a pink pentagon. Lactose, the sugar found in milk, is formed from one glucose and one galactose unit, with glucose shown as a blue hexagon and galactose as a green hexagon. Simply put, disaccharides are double sugars, created by linking two single sugars, that is monosaccharides, together. Similarly, polysaccharides are formed by continuously linking many monosaccharide units together. Disaccharides are made of two monosaccharides linked together, while polysaccharides consist of long chains of many monosaccharide units. Polysaccharides are complex carbohydrates made up of hundreds or thousands of monosaccharide units, usually glucose, joined together by glycosidic bonds through condensation reactions, where water is removed each time a new unit is added. They serve two main functions: energy storage, like starch in plants and glycogen in animals; structural support, like cellulose in plant cell walls. Their structure, whether branched or unbranched, determines how they function in living organisms. At last, the diagram shows three types of polysaccharides, which is starch, glycogen, and cellulose, and how long chains of glucose are arranged in each. It shows a small part of the glucose chains and then how the whole structure looks. You can see that starch has chains with some branches, whereas glycogen has many branches, and cellulose has straight, unbranched chains that pack tightly together. This helps us see how glucose chains can be linked in different ways to make these different molecules. In summary, we can say that polymers are large molecules made by linking many smaller units together, creating diverse structures and functions.